So we went all through this, right? I don't remember. It was a long time ago. Anyway, highly stereotyped fixed action pattern, highly flexible, innate learning. Okay, so as we remember from birds, remember birds? Birds. This is a bird brain. See that? I don't know if you can comprehend that because it turns out that our brain size has been getting smaller in the last 20,000 years. We're not as smart as as the cavemen were, it turns out. Anyway, it's about neurons and the proximate mechanisms of behavior in terms of neurons. So here we have fixed action pattern. I know I went over this. And then in terms of the stimulus, obviously this guy gets the best. This is pretty good, but they really respond actually to the bigger the red, the more release they get. And then it goes to circuits in the brain, and lo and behold, the chick pecks. And then that is in turn a releasing mechanism for the adult to regurgitate food. And all this is, yeah, I know we've done this. Okay, so unlike human males, these guys will mate with anything, even an inanimate object. Of course, I forgot about Barbie. She's inanimate, but for some people, she's real. If you put Siri's voice over Barbie, then you've got, something pretty good. Okay, I talked a little bit about this. This guy is deceiving. These are blister beetle larvae. Deceiving this, releasing a pheromone. They come in, they all climb on board. He goes and mates with a female. They climb on her. They go back to the nest and eat up all the eggs. An evolutionary arms race between bats and moths. We've been following this kind of bats and moths interaction for a while. There's a lot of time dedicated in the book to it. But they've been finding some stuff recently. So an evolutionary arms race is like the Red Queen hypothesis. It's antagonistic coevolution. In this case, the bats are trying to catch the moths. They've got a pretty good method of doing it. They're using kind of echolocation. Of course, from the moth side, they want to avoid it. And so the moths have a number of ways to avoid bats. And in turn, in some bats, the bats have then run a counter defense against the moths. So for the bats, we have echolocation of moths. The moths can detect, they've got ears on their wings or underneath on their thorax, and they can detect bats echolocation from like 30 meters away or more. And if a bat is echolocating from there, they tend to just try to serendipitously fly off to get out of the way. But the louder and nearer bat echolocation triggers this FAP evasive maneuver, and we'll see how it actually works. And then finally, moths are able to jam bat sonar by emitting an ultrasound of their own. Now, the bats are not only in this interaction with moths, but they also have an interaction with each other. They're in competition for food. So, in terms of bats are actually also jamming the ultrasound of other bats competing for moths. So they jam the bats kind of like what the moths did, and then they swoop in for the meal. At the same time, the moths, of course, are they're sending out ultrasound signals too to deter the bats from actually locking in on where they are. And then finally, because these moths are able to do various things, they found some bats that kind of they still use echolocation, but it's a stealth. It's a very quiet, low-intensity ultrasound. The moths really can't detect until the bat is about 3 meters away. And then, of course, they lock in and they go in for the kill. So, this is exactly what the Red Queen predicts. As one side gets better at what it does, the bats in terms of capturing moths, the other side has to keep up just to stay in the same place. The moths have all these mechanisms that they then evolve to deal with the bats and to avoid getting eaten. And then in return, the bats are finding new ways so they won't be detected, and the moths can't actually deter them. Okay. So this is the bats and moths in action. Very nice technique. Oops, got away. Okay, so on to the next slide. Well, I'll give you a kind of a run through about what's going on here. The bats emit this ultrasonic call. They get echoes back. They locate prey. That also, the moths can hear that. So they've got this very simple circuit that triggers an escape behavior, which we'll see kind of in a second. So the moths detect the bat call, then they take evasive action. If the source is distant, they kind of fly away. It's much more dramatic. It actually happens where the echolocation from the bats actually triggers an automatic response in the nervous system. Here we've got our noctuid moth. Got a brain up here, thoracic ganglia, and then these ears, 2 on either side. And then we've got this tympanic membrane. It's a mechanoreceptor, so you get echolocation. It makes it vibrate. It's then picked up by the A1 and A2 receptor cells, and then that signal is sent to these interneurons. We'll get into it. There's a nice little chart that's coming up. So Neurons and their operation. Let me get this. So there's a dendrite, and the electrical charge is initiated from there, is passed on from another neuron, goes down all the way down to the next axon. So this is the direction of the action potential. The action potential does change in electrical charge, and it allows communication between and signals between the two sort of neurons right in here. So if the signal isn't strong enough, the dendrites pick it up from the previous cell, then it doesn't create a strong enough action potential, which addresses the permeability of the cell membranes, and it's the way the ions actually move through the membranes. But if it's strong enough, it's sent all the way down, and then it, of course, there's neurotransmitters that go through the gap between these two cells here, and that triggers the action potential if it's strong enough in the following cell. And so there's this kind of volley of cells, neurons, cells, action potentials onto the next cell. Neural network of the moth. And as you can see, we've got these ears on either side of the body. Those are the tympanum membranes. And then there's these sensory neurons. There's A1 and A2. The signal is then passed on to these interneurons. So the interneurons are part of the thoracic ganglia, and that signal is then transferred to the brain. At the same time, the signal then is transferred across back from the brain down to the thoracic ganglia, other interneurons, to the motor neurons, which then affect muscle wings. And in terms of evading, so this only happens when the bat is very close. So in terms of evading, when there's really strong signal from the A1 receptors, that triggers a response where it kind of, I guess it stops the signal, or it changes the signal from the brain, and the muscle neurons don't work in synchrony anymore. And that causes the moth to kind of go off in any random direction or fall down or whatever, where the moth doesn't even know where it's going, but neither does the bat. Okay, properties of the ultrasound detecting auditory receptors of the moth. So here are these different receptors, the A1 receptor, the A2 receptor. When there's low intensity stimulus, the A2 receptor doesn't respond at all to the sound, and the A1 receptor doesn't respond much too. However, when there's moderate intensity stimulus, the A1 receptor fires, and the A2 receptor doesn't. And then finally, with high intensity stimulus, we've got a situation where both actually fire due to these ultrasound pulses. So you have these ultrasound pulses right here, all the way down the line. The A1 receptors are obviously firing. In contrast to pulses, which the A1 receptors obviously respond very strongly to. If there's a kind of a constant sound, they initially respond to it, and then the receptors kind of stop responding to it. So it's the pulses that are part of the stimuli that the A1 receptors are very sensitive to do. There's no pulses, which correspond to the echolocation pulses of the bats, then it's kind of like the receptors, in a sense, ignore it. Moths have different responses depending on where the bat actually is. The A1 receptors are pretty sensitive, even at low intensity, but certainly at moderate intensity. The moths can detect bats from 30 meters away, maybe further. The intensity of the echolocation is kind of proportional to how far away the bats are. So far away, the intensity is much lower. The A1s don't fire very significantly, but they certainly respond. And then as the bat starts to get closer, they can tell where the bat is actually coming from because, of course, they've got ears on either side of their thorax, and depending on which one is facing the bat or not, they can determine where the actual bat is coming from. The moths can also detect whether the bat is above them or below them. Of course, the wings kind of get in the way of or block the echolocation, so when that happens, they can kind of tell, especially since the wings are going up and down, there's kind of a modulation, and the moths can tell whether a bat is then up. If the bat is underneath, there's intensity all the way, and they can tell then it's underneath. Therefore, the moths can take evasive action. Let's say from 30 meters away, they determine where the bat is, and then they can respond in turn by flying away from that bat. But as a bat actually hones in on a moth, and of course then it's sending these echolocation pulses, and they get more rapid, the pulses, and stronger, the A1 receptors really start to fire, as we saw earlier. And at a certain point, the A2 cells also start to fire. At this point, the bat is perhaps less than a second away from eating the moth, and this requires extremely fast evasive action. So when the A2 receptors fire and the A1 receptors are firing, the message goes to the brain, and it seems to shut down the central steering mechanism, which causes the moth's wings to beat out of synchrony or irregularly, and it causes very erratic or unpredictable flight by the moth and the bat. The moth, of course, doesn't know where it's going, but neither does the bat. And of course, so these signals go back to the interneurons, then to the motor neurons, and then that are attached to the cells of the muscle cells that cause various flapping of the wings. In the following video, we'll see how this actually works.
This yellow underwing moth is very unlikely to end up as their prey. It's equipped with an early warning system to detect a bat attack. Bats hunt with sound. It gives them a better picture of their surroundings than their eyes can see in the darkness. They produce very high-pitched squeals beyond the range of human hearing, and then listen for echoes bouncing back off potential prey. Each echo gives the bat a single audio image, like a snapshot of objects close by. Repeated squeals give the bat a more continuous impression. Once the prey is in range, the bat locks onto the target and homes in for the kill. But this moth can hear what's coming. Its ears are specifically tuned to the bat's sound search system. The moth's ears are simple pits just above its legs, hidden under its fur. The bat's shrieks vibrate the moth's eardrums, triggering a reflex action of nerve impulses, which fire straight into the wing muscles. As the bat closes in, it squeals higher and faster, and the moth's wings react, changing its flight path to duck out of danger. So the moth's life is saved by a dramatic nosedive, switched on entirely by sound.
Moths also have another counter-defense or defense against bats. They can actually jam a bat's echolocation ability, and so it explains this in the following video. So we've seen what moths can do to bats, including taking evasive action or jamming their echolocation. But bats also have to contend with other bats. They're all hunting for insects for food, and there's a lot of competition. So when you see all these bats come streaming out of a cave, there's a lot going on. Well, it turns out bats can also jam bats, so here's a video on that as well. Now that we've found out what bats and moths can do, it turns out that humans also have echolocation abilities. It's certainly not as refined as either the bat or the moth, but this following video will show you this fellow who's blind, who can actually use echolocation using clicks that he makes to navigate the world around him. And he's taught this to many different people. As you know, many animals are able to detect things that humans can. For example, ultraviolet light, electrical signals, heat, like pit vipers, things like that. So here we have a good example. A lot of flowers signal insects or insect pollinators that they are good to eat and kind of guides into where the nectar is. Another interesting ultraviolet detection system is with kestrels and voles. Now kestrels, which we have right here, are very small falcons, and they can eat all sorts of different things, including birds, but also these little voles. Okay, and voles live in these fields. The question is, for the kestrel, how does it know which fields have lots of voles and which fields don't? Well, it turns out that when kestrels fly over these fields, and there's lots of voles, there are vole runways all over the place, and on those runways, which are just not below the surface, but on top of the surface, but underneath the grass, voles, they defecate and they urinate. And that stuff actually shows up under ultraviolet light. So kestrels can actually see ultraviolet light and see those feces and urine. And we can see that right here. This is under ultraviolet light. So when a kestrel flies over a field and there isn't much of this ultraviolet signature of feces and urine, then it keeps flying and it finds another field, and if there's a good signature, then it knows there's lots of voles there, and it's a good place to hunt. Monarchs, butterflies, also use UV radiation in terms of finding out where they're going, as well as polarized light. So if we look at this map right here, from the Rocky Mountains, there's a population throughout here that migrates to the coast and hangs out. And then there's these Midwestern and Canadian populations that migrate back down to Mexico. And this is a multi-generational migration where the ones that migrate down to Mexico overwinter, then migrate back, lay their eggs on milkweed. That generation hatches, flies up to their summering grounds, and then depending on how far up they are, they either lay another generation that then flies back down, or they themselves fly back down to Mexico. So it turns out that monarchs use radiation, UV radiation, to actually begin their journey, and then they actually switch to polarized light. And they found this out by using polarizing filters as far as the polarized light. So, for up or down filters, or no filter, they were able to see the polarized light as it is. So these are directions along the x-axis, and then number of events on the y-axis. But if they put the horizontal filter in, then they were thrown off as far as which direction they were actually going. When moths detect bats, as we saw, it's pretty much a one-step process. It's very simple. You have those A1 receptor neurons that are activated by the tympanic membranes. And then they go to interneurons and whatnot. And that's fine for what they have to do, but some pattern or some behaviors are a lot more intricate and require more intricate signaling. And one of those is what are called central pattern generators. So, as we can see, some require a series of muscular responses that are not under the command of a single cell. So the question is, what is a central pattern generator? Well, you can see neural clusters play a kind of a pre-programmed set of messages, kind of a motor tape, to organize the motor output underlying behavior. So once it's activated, that motor neuron stuff tape plays out all the way till the end until the stimulus that was causing the behavior ends. Central pattern generators are rhythmic patterns of behavior, and they're called oscillators. And they include flying, walking, breathing, things like that. When they're activated by a releaser, these controlling neurons make two cells fire in opposite phases by alternating excitation and inhibition. Tritonia are these little sea slugs or sea hares right here. And they're not necessarily very smart, but they don't have to be. But this one looks like it's in trouble because if it doesn't move, there's this sea star that is probably going to latch onto it and eat it. Well, once the Tritonia actually senses the sea star, or any other predator for that matter that's slow moving, the central pattern generator, so we can see up here we've got sensory receptors, and then we've got swim initiation neurons, so excitation and inhibitory, and then central pattern generator is right here, and finally flexor neurons that make these muscles fire. So it requires a lot of coordination and between many hierarchically arranged neurons, as we can see hierarchy right here, causing muscle contractions where it can kind of swim away, as we'll see right here. Here's the video that we're going to play on this. What is stimulus filtering? Well, organisms, including us, get bombarded with all sorts of information that's coming in. Some of it's relevant, some of it's not. Some is very important to pay attention to, others less so. And so our brain actually filters a whole bunch of information that we just don't really care about and that really isn't important to our, let's say, survival or reproduction, whatever. So stimulus filtering is the ability of neurons or neural networks to filter out vast amounts of information in order to focus on the biologically relevant ones. Stimulus filtering takes place at the level of the sensory receptors, the interneurons, the brain, all of that stuff. I put it on and left it on. Why did it do that? Okay, anyway, damn. For example, here we have this fly, Ormia ocracei, is attuned to the frequency of a male cricket's call, which is around 4 to 5 kilohertz. So the cricket is actually, the male is calling, of course, for a female, and the fly, which is a parasitoid fly, it's going to lay its eggs on the cricket, follows this signal. The male is hoping that a female will come in. Instead, we've got this fly that comes along and lays its eggs. So we can see the frequency along the x-axis here, which is about 4 to 5 kilohertz. Oops. 4 to 5 kilohertz. And then the threshold for the fly's response, which is along the left side y-axis, and the relative intensity of the cricket song on the right. And so as the male, as the cricket song is being made, the female fly, who's going to do the egg laying, not the male doesn't even matter, corresponds with the cricket song and those 4 to 5 kilohertz. So the fly is able to lock on to the important information and kind of get rid of all the other stuff. Okay, so there's this animal called a star-nosed mole. They're found in North America. You've probably never seen one because they spend most of their life underground. And they've got this appendage for detecting prey. And it's amazing. There's, I think, 27 different arms of this. And it's very, very sensitive to any kind of movement or tactile signal that they get. And so there's this one 11 here that's corresponds to this area right here. And this is actually what it looks like. Hold on a second. There we go. So, and this is number 11 here. And then if you look at appendage number 11 and then average area of cortex per input fiber. So essentially, this whole thing is mapped out. This is the actual cortex in the brain. This whole thing is mapped out right onto the cortex. So we can actually see what area is being tweaked or getting a signal and how the firing works. And it turns out it's pretty effective.
In North America, another mammal has paws that look like flippers. These help it to swim under ice to collect insects, but this is not their primary purpose. This creature is a digger, a star-nosed mole. Its paws are spades for pushing aside soil while it tries to locate its prey with its astonishing nose. This has 22 fleshy arms. Each is so densely packed with nerve endings that the mole could touch a pinhead with its nose in 600 places at once, allowing it to locate the tiniest of prey. Living in soil rather than sand, this mole can dig proper tunnels. It constructs a labyrinth of passages and regularly patrols them to collect any prey it drops into them.
Juicy. Gollum.
The star-nosed underground is largely beyond the reach of predators.
Not entirely, of course. Snakes and whatnot, but pretty much. Okay. So, this is kind of a sensory analysis for 4 different insectivores, and then us, and a few other things. So we've got the star-nosed mole. That's right here. As you can see, it's got kind of big digging paws, so there's a lot of cortical material that's dedicated to that. And then, no, actually, this is it, sorry. And then this is the appendages right here. So it's almost like the whole animal is kind of dedicated to that movement and finding prey. Obviously, it changes, it differs. So African hedgehog, I guess, which one is that? I think it's this one. In almost all of them, it seems like the mouth is a big part of it. If you look at humans up here, so our hands have a lot of receptors. If you've ever been injured your finger or whatever, it's usually much more painful than some other part of your body. And your feet, because we're always walking around. Let's see. Well, also there's, what is it? This thing right here. That's, we're always concerned about that as well. So this is actually how our brain is kind of mapped out in terms of our physical world around us. And you can see that there's certain areas that are more dedicated to certain things. So obviously language for us, if we could actually map that out, there would be an area of the brain that's really dedicated to language acquisition and then language use. Oh, shoot. Hold on a second. I don't
Click on the X in the square.
The red one.
The red X.
This one?
Alex,
No,
What, man?
it's not working. It's okay.
whoa, that's right. You're
Usually there's
Alex can answer different questions about the same object.
Come on, how many corners? What shape? What shape? Corner. Four corner, good boy.
Alex hasn't just learned to say
You
a
gotta tell
certain
me.
word when he sees a particular
How many?
object. He's
How
paying.
many?
What's going on here? Come on, you can do it. Okay, so these are New Caledonian crows. They they're on this island in the South Pacific off of Australia, and they make a lot of tools to use to fish out grubs and things like that. Yeah, I don't want to lose that stick. Okay. So these are smart. And then here's a little abstract thinking. I think you saw this before, but just in case.